multiple sequence alignments. When we align more than two sequences to each other, we call it a multiple sequence alignment. Here's an example. It is the 91st positions of a multiple sequence alignment from several different organisms, consisting of the acidic ribosomal protein P0. You will note that in this alignment we have now very different organisms from very different areas of life, and they will appear similar because they have the same origin or genetic origin, and the phylogeny just plays out in such a way that many of the columns are conserved between the different organisms. And as well for many of the columns, even though they are not perfectly conserved, you will see that any replacements are done with similar amino acids. Look, for instance, at sequence column number 70, and you will note that the arginines are sometimes replaced with lysines, both positive amino acids. And similarly, you can see columns of blue, where or light blue that is, where you see that different hydrophobic residues are placed with each other. So this is just a very pleasant way to discover what's conserved and not conserved in different sequences. How do we construct multiple sequence alignments? Well, in principle, we can use the same type of dynamic program as for pairwise alignments. However, we would need to expand the number of dimensions in the dynamic programming algorithm, this would now be, the number of dimensions would be proportional to the number of aligned sequences. So in practice, this is slow for dimensions over 3, 4. So most commonly, we are using other types of heuristics to sort of circumvent the fact that we can't do the full sequence alignment for a large number of sequences. One common such approximation is so-called progressive alignments. This is an algorithmic approach where we firstly start by calculating pairwise scores of all your sequences that you want to align. Then you align the two most similar sequences, and then you replace the two aligned sequences with the alignment. And then if there's still remaining more than one sequence, then you reiterate from point number 1. So here's an example. You want to align 5 different sequences, and you start off then by making a pairwise alignment with all sequences against all sequences. You might end up seeing that a couple of the sequences are more similar to each other than others. So in this case, the green and the blue sequence seem to be most similar, and hence we can use this information to build now as an input, then just align the green and the blue sequence to each other. This will introduce a couple of inserts and deletes in the sequence alignment, but now replace the green and the blue sequence with this alignment. So we will have now, instead of a 5 sequence, we have 4 sequences. So now next step is now to see which is the next most similar sequences. Actually, we can recalculate the tree here, and there's different heuristics how to do that. Often we don't need to recalculate the full tree, but you can just sort of replace the distances to the combination of the green and blue sequence with the average of the distances to those two. Next most similar sequences are the magenta and the red sequence, so we align those two. And so now it remains 3 different sequences. We take those as input now and make a new sequence, new pairwise distance calculation, and see that the combination of the red and magenta is closest now to the yellow sequence. So we align that sequence, and we replace those 2 sequences with 1. Hence we end up with now just having the combination of green and red, green and blue, and the combination of red, magenta, and yellow. So we align those two to each other, and then we end up with the final sequence alignment. So this is called progressive alignment. It has a couple of features, and one of the unfortunate features of this is that once you've made a pairwise alignment, the alignment itself is fixed for the next iteration. So if we would find out that there's sort of more optimal approach to align the green and the blue once we confront the other sequences, that will not alter the alignment itself between the green and blue. They will be kept fixed into each other. There's multiple different heuristics, and they are often using this progressive alignment. They have other twists to it as well. But here are a list that I've sort of kept. It's K-align and MAFT, Clustal Omega, Muscle, T-Coffee, and ProbCons. K-line is a really fast, and so it's suitable for larger alignments. MAFT equally is suitable for medium to large alignments, and Clustal Omega the same thing with that. Clustal Omega has a sibling, or rather an ancestor, in Clustal W that you see used just everywhere, including our book, unfortunately. Never use Clustal W. It's an old outdated method. And then we have Muscle as well. It's accurate for medium-sized alignments. The other two methods here, T-Coffee and ProbCons, they're very accurate. Unfortunately, they're as well quite slow. So I could highly recommend those two, but you should use it for smaller alignments. Thank you.